going to shift gears a little bit and really talk about this from a little more academic perspective and also from a more patient care perspective. David Solit, I'm the director of the Kravis Center at Memorial Sloan Kettering Cancer Center. I'm a physician scientist who sees patients, but also run a discovery research lab. I'm also here representing AACR as a member of their publication committee and one of the senior editors of Cancer Research. So what do I mean by precision medicine? As you've heard a little bit, it's trying to match up the right drug for the right patient. But more than that, it's actually an effort to try to develop new therapies. And really, our paradigm for developing new therapies in this context is pretty simple. What we try to do is we try to define new drug targets. These are usually genetic alterations that are present in a patient's tumor, but are absent within their normal cells. We then develop drugs that inhibit those targets, those usually mutated enzymes within the cancer cells. And then what the real challenge has been to really move this field forward is being able to identify which patients have a mutation in a particular drug target so that we can match them up with the right drug. And so how do we identify these targets? One way we do this is there are some international efforts ongoing to take thousands of tumors from patients with particular cancers and really sequence their entire genome. So that's really a retrospective discovery effort. But I was asked to talk briefly about a methodology that we've been using in my laboratory group that we've really pioneered to really take the opposite approach. And this is called an extraordinary responder approach, where we take patients who have responded to cancer drugs that are either approved by the FDA or drugs that are in clinical trials, to which most patients didn't respond. So these were the minority of patients who benefited, and try to figure out what made them unique so that we could identify what the marker was of sensitivity in those patients, and then maybe salvage drugs that otherwise would have been sort of tossed out as ineffective, but try to now focus them down onto the subpopulation of patients who are most likely to respond. And so the first example of this was a trial that we did a few years ago, back in 2008, and this was a trial in patients with bladder cancer, and it was of a drug called everolimus, and this is a drug that's now approved for use in patients with kidney cancer and breast cancer, but we were trying it in patients with bladder cancer, and unfortunately, like every clinical trial that we've done in bladder cancer over the past 3 decades, this was a negative study. So if you look statistically across the entire population, there were 45 patients on this trial. Almost all of them did not respond to the drug, and there didn't seem to be any difference in their survival as a group compared to historical patients with bladder cancer. But there were 2 patients on this trial who responded, and I'm just showing you one of them and just showing you the dates of some of her CAT scans here. And I don't seem to have a pointer, but oh, here we go. So I don't know if it'll show up on the screen. It won't. She actually presented back in 2009 with actually cancer of the bladder, which had spread to her lymph nodes. And what we try to do in this situation is we give these patients chemotherapy. We try to shrink down the tumor, and then we take out the bladder and all the sites of disease that we can find. And if we do that in a minority of patients, we actually cure their cancer. And this woman was briefly without evidence of cancer for a few months, but unfortunately by January 2010, her cancer had come back. And the red arrows there on that first CAT scan are showing this metastatic cancer that she had developed. Now, in this situation, in this type of bladder cancer, there's no standard of care. Patients live only on average about 9 months. And so we encourage these patients to go on clinical trials. And this woman went ahead and enrolled onto this everolimus study that almost nobody responded to. But by April of 2010, she had a near complete response. By July 2010, a complete response that was durable July 2011, and then off the slide 12, 13, 14, 15. She's in fact still on this drug, now over 5 years, with a complete response to this drug. The other 44 patients on this study have actually died of bladder cancer. So she's this unique outlier who benefited where nobody else did. So what really made her unique? Well, you've heard about whole genome sequencing. This was actually the first bladder cancer which ever underwent whole genome sequencing. And what that means is we sequenced every single DNA molecule in her tumor cells, all 3 billion base pairs, and then took several months to analyze this whole genome. And what all that work showed us is that she had 2 mutations in 2 genes, one called TSC1 and another called NF2, which we knew, when inactivated, caused activation of a protein called TORC1. And the reason that that's important is that TORC1 is the direct target for this drug, everolimus. So in retrospect, this woman was the perfect candidate for this drug. We just didn't know it going into the clinical trial, but one could imagine that if we knew that information before the clinical trial, maybe we would have done this clinical trial a little bit differently and maybe had enriched the clinical trial for people who were more likely to respond. So we've done the same extraordinary responder paradigm with other patients. This was a clinical trial of patients with ovarian cancer, just showing one example of a patient on that study. It was run by a cooperative group called the Gynecologic Oncology Group, and tested a drug called a MEK inhibitor in patients with recurrent and metastatic ovarian cancer. And essentially, there was only one patient on the study who had achieved a complete response. There was some also partial responses on the study, and this is the one patient who had a complete response. And they didn't understand, at least at the time, why. They had looked at some older assays looking at specific gene mutations, could not figure out the reason for this. And I would just point out that this response is now ongoing over 6 years in this woman who really was out of therapy. She had been through 3 prior surgeries for her ovarian cancer, 3 prior systemic agents, and really had no other options at this point, and has now been 6 years cancer-free on this drug. So what made her cancer unique? Again, by doing this next-gen sequencing, looking at hundreds of genes at the same time, we were able to figure out that she had a deletion in a gene called MAP2K1. And the reason that this is interesting is that MAP2K1 encodes for a protein called MEK1, which is actually the direct target of this drug. We just didn't know she had a mutation in this gene going into the clinical trial. So again, same case as the first case. We were able to retrospectively solve this extraordinary responder, but maybe if we knew that information prospectively, we could do more rational clinical trials. And I just want to point out, we always go back to the lab and actually prove this when we do these retrospective responders. We go back and we show that this mutation is an activating mutation, turns on the protein all the time, and that this drug actually inhibits that activated mutation. So this is really the paradigm. We take these extraordinary responders from these failed clinical trials. We take tumors from those patients. We do genetic sequencing. We figure out what we think made them unique and why they responded. We always go back to the lab to actually prove that biologically. But then what we really need, this is really the greatest need, is we need an assay which will allow us to look for these same mutations in other patients so that we can do a second clinical trial. We don't want to change practice from a single patient. What we want to do is a second clinical trial where we enroll patients who have the same genetic mutation and ask, well, how often do they respond to that same drug? And we don't expect necessarily all of them to respond, because some of them could have a complement of co-mutations that make them resistant, but we need to answer that question, how often do they respond, if we ultimately want to change the standard of care. So how are we going to do this at Sloan Kettering? The way we do this at Sloan Kettering is we use a next-gen sequencing assay called MSK Impact. What it does is it tests for these mutations in 410 cancer genes. We actually take all that information prospectively, we put it into a database. That database is accessible in a HIPAA-compliant way to any researcher at Sloan Kettering, and that database is also linked to clinical information on these patients. And I'm just showing one screen shot from that database, showing that there's a host of patients who have these MEK1 mutations, the same type of mutations we saw in that ovarian cancer responder. And what we go ahead is we take these mutations, we actually make them in the lab, and we prove, okay, this mutation's real, it's activating, it would be a good one for a clinical trial, or maybe this other mutation's what we call a passenger mutation, meaning it's a mutation in the gene, but it has no function, it's not activating the gene. And so once we have this assay, what we need is then clinical trials that will allow us to efficiently test these hypotheses, which is, if you've got this mutation, do you respond to a particular drug? And really the concept that we've been pushing in part is a study design called a basket study. And so most clinical trials historically have been focused around particular disease types, say a clinical trial in breast cancer, or a clinical trial in ovarian cancer or lung cancer. And these type of trials really turn that paradigm upside down. And instead of focusing the trial on a particular cancer type, they focus the trial around a particular gene or gene mutation. And this is a clinical trial that I run. It's an international study. There's about 30 centers participating in this study, where we are looking for patients who have mutations in a protein called HER2. You heard about it a little bit earlier. We know that gene amplification is a good target for HER2 therapies in breast cancer. We're actually looking for a different type of alteration in HER2 called a mutation. And so everyone with a HER2 mutation is eligible for this trial. It doesn't matter if you've got bladder cancer, colon cancer, endometrial cancer, gastric cancer. We always have an other arm on this study, and they're all eligible. And what we're asking is, if you have this mutation, how often do you respond to this drug called neratinib? And does that response vary as a function of what type of cancer you have, say breast versus ovarian versus bladder cancer? And we've seen some dramatic responses on this study. So this is a patient with what would be called, quote unquote, HER2 normal breast cancer, because they don't have HER2 gene amplification. But this patient has a mutation in the HER2 gene called V77L. And you can see on the left her pre-treatment scan showing all of this disease in her bones and other soft tissue by this PET scan. And you can see within 2 months, all of that cancer had gone away. And this is not really that conceptually novel from a biology standpoint. We've known that HER2 is important in breast cancer for 20 plus years at this point. We only knew that these mutations existed for the past few years. And the question has been, how do you find these patients with these mutations to really test this hypothesis? Because the key point is at the bottom of the slide, which is that even in 2015, essentially all patients with this particular mutation in the United States don't know that they have this mutation, because they've never, in fact, been tested for this mutation. So that's really the major challenge. How do we test for these mutations? How do we find them so that we can put these patients on clinical trials and prove drugs like this work? And even if we had the test in place, how do you bring these therapies to the patient? This trial is only open at about 15 institutions in the US. And so if you have this testing at a place where the drug's not available, you don't really have access to the therapy anyway, so was it worth doing the testing? So these are really the issues that we're working with. Now, just one more data slide, and I just want to point out that this is not just for these targeted kinase inhibitors. This was a study presented at last week's ASCO meeting, the American Society of Clinical Oncology, and published last week in the New England Journal, showing that if you've got a pattern of mutations in your tumor called MSI or MMR deficient tumors, it's a type of hypermutation that we see in a diversity of tumor types, in particular colon cancer and endometrial cancer, that if you have that pattern of hypermutation, not a specific mutation, but a general pattern, that you respond incredibly well to immunotherapy. And so, again, we're not just predicting with these tests response to kinase inhibitors, these small molecule pills, but also these newer immunotherapies that are using the body's immune system to fight off the cancer. So I'll just leave my conclusions here. It's at least my opinion that genetic analysis of all patients will become routine in the near future. This is really being held up by issues like cost, what assays should we use, how is that going to be regulated. This type of testing is already standard of care in some cancers. Those include lung cancer, colon cancer, melanoma, but not things like breast cancer and prostate cancer and endometrial and ovarian cancer. So for many tumor types, no testing at this point is considered standard of care and reimbursable by insurance. And I really think that once this testing is more broadly available, it's going to improve patient outcomes by, one, optimizing treatment selection, and by, two, really accelerating the development of these novel therapies, which have been slowed down because we can't really match up the right patient with the right drug. So thank you.